So, to make introductions,
Yeah.
I'm Matthew Holma. I'm interviewing Dr. Jack Tuszynski at the University of Alberta. He also has a joint appointment at the University of Turin in Italy, and yes, he has a broad-spanning field covering. He started in physics, condensed matter physics, if I'm correct, and moved on to oncology and drug design, as well as forays into the nature of consciousness. And I was just wondering, Jack, if you could briefly describe what made you get into biophysics to start.
I see. Well, I was always interested in biology, but I think for the first decade or two of my scientific career, biology was very qualitative and not enough quantitative data were available. And being a physicist, for me, the term model means a mathematical predictable model, something that you can test against future experiments. And until maybe late 90s, there was really nothing in biology that was so amenable to mathematical/physical modeling. And this changed as biology entered into nanoscale, cellular and subcellular areas where precise measurements were made and data were produced that could be analyzed mathematically. And my first and still major interest was actually the cytoskeleton, polymerization processes of microtubules mainly, but also actin filaments and the motor protein processivity, the machinery of living systems, and my first interest was to try and make it mathematical, make mathematical models of these subcellular processes, and that gradually grew into more and more detailed representations of structures inside the cell at the level of atomic resolution, and that is, of course, where physics reigns supreme.
Okay.
So that was how it started. And I have to say that my transition period occurred when I went on sabbatical to Germany in early 1990s. It was my first sabbatical, and I used the time to rethink what really made me excited about science. And I visited a lot of places. I was based in Düsseldorf, which is close to the Dutch border, and I used to go shopping to Maastricht on the other side, or to Belgium, not too far. Belgium, the Netherlands, central located, and I went to places like Oxford and Paris and all around Europe, in fact. And I thought for a year about what to do, and I decided to move into biophysics from condensed matter physics, because to me that was actually more exciting. There were more opportunities, and when I came back from sabbatical, I went straight to the Department of Physics office to speak to the chair of physics. And at that time I was a full professor already, so I could do what I wanted more or less with tenure. So we have academic freedom, luckily. And I told him that I intend to pursue biophysics and would like his help to set up a group. And that was about mid-1990s, and finally, after 2 decades, we now have a group of biophysics.
Mm-hmm.
But at that time, this was frowned upon, and in fact, I was given a cold shoulder.
Okay.
The department and the chair, even the dean of science, who was a biologist, didn't like the idea.
Yeah, yeah.
But I will not name names.
Yeah, probably wasn't considered real physics.
It was considered, for the longest time, biology was considered, how shall I put it, gently, pseudoscience, or not quite a mature branch of science.
Yeah.
Now it's quite different, I think, and the problems in biology are much more complex and require a lot of intellectual rigor, different skill set than in physics for sure, but they are not easier to solve, as you probably know.
Yes, yes.
So this is how it all started, and I was looking for a place where I could do physics and biology, and physics at that time wasn't too keen. But an opportunity arose, and I reached out to some colleagues in the Faculty of Medicine, biologists, and also at the same time in mathematics there was a big move towards biology. So there's a mathematical biology center here at the U of A, which is doing some very good science. Mark Lewis and Gerda de Vries, Thomas Phelan, and people like that. So I think I was able to find some kindred spirits around the campus. And eventually, as you know, I moved to oncology to work on practical problems in terms of drug design for chemotherapy. And now I'm going back to physics.
So I guess, what was your initial entry point into biophysics? It eventually became drug discovery.
Yeah, but the my initial work in biology was actually two different entry points. The first one was polymerization of protein filaments, so microtubules, actin filaments, writing differential equations that describe these processes and solving them and fitting to experimental data. Even eventually, I wanted to do some experiments, so I found a biologist on campus who came from Harvard actually, and was very well trained in microtubulin purification. And we went to get some cow brains from a slaughterhouse and extracted tubulin from cow brains and did some experiments just for fun. So that was one, and the other entry point actually was interestingly quite different from that, and that was also accidental.
So, sorry about that.
Yeah, no problem.
We were just talking about pharmacokinetics and pharmacodynamics and the
Correct.
distinction between those.
Yeah, should I repeat that? Or?
No.
Okay. So that's the second entry point into life sciences for me.
Okay. And from there, how did you get into consciousness research?
Okay. Well, that's another interesting story, and maybe not most, but many things in life happen by accident. And in this case, it was actually unintended consequences of attending one of the most amazing conferences I ever attended in my life. And I was invited to actually co-organize a conference on which was called What is Life? And that was a NATO-sponsored, believe it or not, conference. And in the U.S., in Tucson, Arizona, the organizers were Stuart Hameroff, Alvin Scott, and they needed and Steen Rasmussen. All of them were from the U.S., and they needed somebody from another country, another NATO country. And Canada is a NATO country, so they found me. So I organized with them this amazing conference, which dealt with the foundations of life, including artificial life. What is trying to grasp what is different between living and non-living systems, and with some fantastic speakers. And at the end of the conference, everybody was excited. This was, by the way, the conference took place during the first Gulf War, January 1991. I remember vividly when we were watching the news from Iraq and bombing and all kinds of horrific things. And that was in the middle of the conference. There was a lot of nervousness. But at the end of the conference, everybody was excited about science and trying to come up with a continuation of this. And we came up with all kinds of topics. And I remember vividly, Stuart Hameroff said, well, guys, this is interesting, but I have something which is even more interesting than that. I said, what can be more interesting than life? And he said, consciousness.
Mm.
So the next conference in 1992, maybe three, I don't remember now, was supposed to be, what is consciousness?
Mm.
And that actually turned out to be a series of annual conferences, which now number 26 or 27, I don't remember exactly, alternating between Tucson, Arizona, and another location around the world, which involved Sweden, Denmark, Austria, China, Japan, and I don't remember other places, but Hong Kong probably. So these conferences are now called Toward the Science of Consciousness, and I was sort of involved from the beginning, and somewhat reluctantly, because I couldn't at that time see a scientific component to it. That's why it's called Toward the Science. We're not there yet, but and that gradually became more and more scientifically well defined. But there's also always a group of people attending this conference and presenting philosophy, even theology, and Eastern meditation, all of the various aspects of consciousness, which we know experience, we do as human beings experience every day, but it's a subjective experience, and that's the difficulty in trying to grasp it scientifically, because science is an objective endeavor, not a subjective, right?
Yeah.
So anyway, that's how it started, and again, as I said, I was reluctant because of these limitations, but somehow I always found incredibly positive and huge response to whatever I wrote about it or presented. There was a lot of interest, and that is gratifying to a scientist when you see your work being appreciated and cited and invitations followed and so on. So that's how it all started, and the entry point again into consciousness is through microtubules, because of the focus that was put on microtubules through the work of Roger Penrose and Stuart Hameroff in the OR theory of consciousness that hypothesizes microtubules to be the site where conscious processes originate or become concretized, if you will. It's a bit more difficult to explain, but maybe later on I'll explain how that comes about.
Okay. Yeah, so if my understanding is correct, Hameroff, he basically, there's a natural experiment that you can do on anesthetic patients who basically lose consciousness. And he was examining the site of action of these anesthetics because it actually wasn't well known, their mechanism of action before.
Well, even now, yes.
Mm-hmm.
And this is precisely the point where science meets medicine, meets consciousness. It's probably the best place where to explore consciousness, because as you know, and as you mentioned, anesthesia is probably the greatest achievement in medicine. Has a century and a half of implementation and allowing us, surgery and dental procedures without pain, and et cetera, et cetera. So it moved medicine incredibly forward, but we still don't know exactly how it works because of the fact that anesthetic molecules, of which there are maybe at least 20 different ones that are being used, they act with different potency, but there is almost nothing in common in their chemical properties. So in terms of well-known effects such as binding to receptors, which is lock and key principle, so shape matching. Anesthetic molecules don't have that. There is no obvious shape matching principle involved in anesthetics. Some molecules which are very similar to anesthetic molecules don't act in anesthetics, and some which are anesthetics, like xenon is a single atom, they don't really give you that simple property that you can latch onto. And so the question is, what happens? Where do they bind? And it's known that anesthetic molecules, their potency correlates with lipophilicity, so they bind into hydrophobic areas, and membranes are natural places where hydrophobic areas exist. But also hydrophobic areas exist in proteins. So there is kind of dichotomy. Most scientists believe that anesthetics bind to membrane receptors, but there is, and this is what Stuart Hammer, who is an anesthesiologist and spent his entire career being a practicing anesthesiologist, but also a scientist trying to understand the origins of anesthesia, proposed that there may be natural places inside neurons where anesthetic molecules may switch off cognitive processes, in other words, consciousness. We know that that happens. And it's amazingly, anesthetics act on humans and animals the same way. So there is no special anesthetic for animals or humans. They all act, even on very primitive organisms, which was experimentally verified, which is interesting. Therefore, it's not about the human brain as a complex structure. It's something simpler. Maybe single neuron could be anesthetized, and therefore you have to look inside the neuron, and we know for a fact that pretty much all anesthetics, I think I would say all, bind to tubulin. That is experimentally confirmed.
Okay. Wow.
Yes. And so that's interesting. And when you look at the proteins which were tested for anesthetic binding, only two proteins appear in all these experiments. I believe it's actin and tubulin. So both are members of the cytoskeleton. So inside the cell you have the complex architecture of microtubules, actin filaments, and intermediate filaments which maintain the shape of the cell and also participate in many processes, including cell motility, so movement, and transport. Everything that goes inside the cell in terms of moving mass, cargo from point A to point B, needs to be transported via these highways constructed from microtubular actin filaments. But there may be even a deeper level, and this is where anesthetics, we don't know if anesthetics, at least I don't know, maybe there's something in the literature, anesthetics stop the transport, that may be also an effect, but we know that they bind to specific locations in tubulin, which are interestingly tryptophan residues that are excitable by photons, or light.
Okay.
And so this leads to this theory that tryptophan networks in microtubules may be the sites of quantum effects, because they are really quantum excitable. Electronic transitions, these are dipole transitions, which can be triggered by light absorption. And then if a network exists, maybe this could be a network of excitable molecules through light, and that can be quenched by anesthetic or interrupted or disrupted even by binding of anesthetics, which we know that they bind very close to tryptophans. So that's basically the sort of molecular level explanation, largely hypothetical, except for the fact that we know anesthetics bind to tubulin, exceptionally among all the proteins, with one more example. So that points in the direction of at least one of the possible explanations. The other is based on memory receptors, which would basically interfere with action potentials, maybe ion channel functioning, things like that. I don't think these two are exclusive of each other. There may be both taking place, but well, we need to do these experiments to figure that out. To my knowledge, nobody has done conclusive experiments on what happens to microtubules exposed to light, and with and without anesthetics.
Okay.
If that could be demonstrated to have a different outcome, then I would say we will be halfway through to solving this enigma.
Okay. That is interesting. That it's a potentially a quantum process whereby these small differences can have a large scale effect on human behavior. I know there's an example in one of Max Tegmark's book where he's in a traffic accident, and if his eye had not absorbed this one photon, then he would have died. But since it absorbed that single photon, he was able to turn.
Great example.
Mm-hmm.
And of course, the question of quantum effects, of quantum consciousness, if you will, was first addressed by Roger Penrose in his books. Oh, yeah. So basically, Penrose gave many examples of non-algorithmic way the human brain works, and pointed out to quantum processes which would solve this problem, where you obtain solutions almost instantaneously by quantum search algorithms. Not algorithm, I use the word which is that's supposed to be. But anyway, quantum search processes, let's say. There are many examples of this. Intuition, premonitions, extrasensory perception, right? And sudden insights, eureka moments, right? Or geniuses, or autism is fantastically fast mental processes, which cannot be explained by simply training somebody, right? Just getting the anyway, there are many examples of this, and that single photon is kind of like that, a flash of insight or revelation or things like that. So quantum, but Penrose at the time when he wrote the books didn't know about microtubules and much about neuroscience. It was just more or less an empirical or observational conclusion, and he didn't have a candidate for how this might work at the quantum level. And so Hameroff approached Penrose and said, "I think I know." So they met I believe in Oxford, and talked about it, and that started their collaboration, which is going on until today, for almost 3 decades.
Okay.
And so microtubules being the most well organized. In fact, I have to say that you asked me about how my interest in consciousness started. It not only started at the end of that conference in Tucson, What is Life, but before the conference started, I remember I came late in the evening to Tucson, flew in, and met with Stuart Hameroff in the hospital where he worked at the night shift. And he said, "Well, I'll see you in the morning, but in the meantime, here's my book, read it." This was a book published in 1987 called Ultimate Computing, which basically laid out the vision of this incredible computational power of microtubules in the neurons of the human brain. It still is, and I have to say, I read it through that night. I couldn't sleep. I didn't sleep 1 minute. It was so amazing. It kind of transformed my thinking about many things. And well, I started thinking about consciousness at that time.
Okay. So I guess there's the view that that microtubules expand the information processing capacity of the brain.
Right.
But there's also some other aspect that's not purely information processing,
Right. Yes. Yes.
or at
Exactly.
least classically.
Yes. So the connection, I guess, from the micro world, microtubule world to our cognitive processes, I think is, as you said, an incredible amplification of the potential, because there's 100 billion neurons on average in the human brain, and current neuroscience views neurons as 0 to 1 switches, right?
Mm-hmm.
And I think it's somewhat derogatory to the neuron to see a living cell as a switch,
Yeah.
as a passive switch, but I think it's not, because we know that living cells have their own intelligence. And I when I give lectures on this, I always direct the readers and listeners to the work of Günther Albrecht Bühler, and he has a fantastic website called Cell Intelligence, where he spent about 2.5 decades of his life examining the intelligent behavior of single cells. A neuron is probably one of the most complicated and powerful cells.
Mm-hmm.
So anyway, now we are looking at a neuron. Inside neurons you have thousands of microtubules, and as opposed to other cells, so people ask that question too, what's so special about the neuron? But neurons, as opposed to, let's say, liver cells, have microtubules which are organized in a stable parallel architecture, parallel bundles, stabilized by microtubule-associated proteins, and also directional. That in axons they are directed in one orientation, whereas in dendrites they are bidirectional. It's basically alternating directions, which may provide some feedback loops. So with thousands of microtubules inside, each microtubule having tens of thousands of tubulin units, each unit could have, as I mentioned, tripped, even if each unit has one quantum state. Now you can see the product of these quantum states exploding exponentially and giving you astronomical potentials for information storage, information processing. We're not even talking about generation of thought or some abstract thinking, but even at the level of just storing. I did the calculation once, and I think I can't remember now the numbers, but it's in terms of just classical computing, they would be the human brain as a collection of neurons with these incredible numbers of tubulins and microtubules would easily outperform a supercomputer like the Blue Gene, running only on about 25 watts of energy, which is the rating of a very weak bulb, electrical bulb, right? As opposed to the Blue Gene, which consumes 1.5 megawatts of power, electrical power. It's comparable to a small town. So outperforming computationally and as also being incredibly energy efficient. That I think is an attractive proposition, why the human brain is so powerful. The processing power, the speed at which these processes occur, we don't know. There are psychological experiments trying to determine how fast we can process information, and they kind of indicate hundreds of milliseconds clocking time. But I think there may be processes which are faster than that, but we simply don't perceive them as such, because there could be subconscious. When we play tennis at the level of, let's say, Wimbledon Championship, or hockey at the Stanley Cup level, the human brain performs incredibly fast, right?
Yeah, the reflex can't be explained in terms of action potential.
It cannot. It's simply impossible. It takes way too long for the action potential to travel, from the receptor and the eye to the brain, then to send the signal to the hand, and all. I mean, it cannot be done, absolutely impossible. I did these calculations once.
Mhm.
But the question is how fast, and depending on how fast you assume it to work on, you have some limitations on, because the power consumption is 25 watts for the entire brain, and only a small portion of it goes into cognitive processing.
Mhm.
Most of it is for maintaining temperature, building structure, maintaining structure, repairing things, and transporting things, and so on. I estimate one estimate is something like maybe 5 watts, which would be like a flashlight.
Wow. Yeah.
That's truly amazing just for the cognitive processing, right? And apparently people who play chess, for example, they consume a lot of energy. So
I have heard that, but yeah, it was Robert Sapolsky.
right. Yes, he's a neuroscientist of great caliber.
Mhm.
Yeah. So maybe you can crank it up, but even if it goes by a factor of 10, you still have a minimal power consumption compared to man-made devices.
Mhm.
So the way it works is that information erasure costs energy. There's a minimum amount of energy,
Mhm.
right? It's the
KB
Landauer.
Lon 2.
Yes, exactly. KT Lon 2. It's the Landauer criterion. And biological information probably is more expensive because
Mhm.
it's not operating on the same principles. It uses biochemical energy, ATP or GTP, and that's more expensive than KT Lon 2. But even then, so then the amount of information processed divided by the time, clocking time, gives you the power. And I think there is still, even when you account for that, you still have possibility of a more powerful information processing output than that of a supercomputer.
Okay. Wow.
Right. So that's and yeah. So the question is where this is happening, and you have to look inside neurons, in my opinion. It's two candidates, in my opinion, need to be left on the table if you want to delve into it. First is microtubules, as I mentioned. The second could still be ion channels, because there's thousands of them and they operate on a millisecond time scale, micro to millisecond, so it could be also used for computational purposes.
Okay. I was curious. This is sort of an area that everybody has thought about to some extent. I was wondering if it affects your regular life in any way, this research.
Well, not every day and every time, but
Mm.
sometimes I think about it, especially when things happen that are hard to explain using linear scientific or Cartesian scientific thinking, reductionism on which we all grew up.
Mm-hmm.
And I think there are limitations to this reductionist thinking. Things like entanglement, quantum entanglement, whether we are quantum entangled as human beings, at least with some other human beings,
Mm-hmm.
where you don't need to communicate, you understand each other, or you maybe are connected emotionally or psychologically or intellectually with somebody who is maybe on a different continent.
Mm-hmm.
So I experienced examples of this in my life, and I think there is something that we still need to explain, which goes beyond mechanistic or classical rules, let's say. So
Mm-hmm.
that's why I think that quantum mechanics, quantum physics offers some interesting possibilities.
Yeah, I do think our models of reality affect basically how everything from how societies are structured to how we think of ourselves. Like, I guess a very basic notion would be atomism, and I think to some extent it does lead to social atomization.
Yes, especially now in this time of COVID-19, I think.
Oh, yes.
It's
It's
very much
taken to the extreme.
Yes. So I think there's still this is fascinating, and the problem I think is that it doesn't really fit nicely into the scientific paradigm. as we started out, it's more subjective than objective, at least at the moment. I would like to do some experiments actually that might shed some light on it, in addition to the anesthetics and microtubules, but also I have one experiment I would like to perform on human subjects, which is along the lines of Bell's inequalities, you know, and other aspects, experiments on particles, entanglement of particles, and demonstrating Bell's inequalities and work, but you have the action at a distance.
Mm-hmm.
You could, I'm thinking about identical experiments on humans, which you could basically examine two humans who are emotionally connected, let's say a couple in love, or parents and children, and then exposing one of these people to some visual imagery, maybe disturbing, and measuring the response of the other person at a distance. If there is some sort of entanglement. It's a little bit contrived, I think, but it might be doable. It might be possible to demonstrate this connection between human minds, human brains, and conversely with random choice of people. That's why here you could have both subjective and objective aspect of it. Two randomly select people probably are not entangled quantum mechanically in any way, but two people who are connected deeply might be, and the question is how far that connection could extend.
Mm-hmm. You need like two time series of some measurable, like fMRI state.
Exactly, something like that. Could be also, you know, our emotional states can be quantified by measurements of heartbeat, or the brain waves, or even conductance of the skin, you know, thermal effect. So you could think of an experiment which is very scientifically appropriate for that kind of thing.
It's similar to Rupert Sheldrake's notion of morphic resonance.
Yes, I agree. And he did experiments, and there's a very interesting movie, I think, made by the German TV, and shows, it's along the lines that actually I explained would be appropriate for humans, but they did it for animals and their owners, pets, dogs and cats, I think, and he went to great lengths to make it objective. That is, for example, instructed an animal owner to meet him or meet somebody at the pub on the way home, and that at that pub he would get the owner would get the message, leave in 15 minutes or something like that. And so nobody, it's like double blind, nobody knew. It wasn't a regular time to return home. It was just something that was instructed from a third party. And they, at the same time, they watched the animal. And there was a camera on that pet at home. And the moment the owner made a decision to return home, the animal got excited. In other words, it showed that there was a connection between the animal and the owner at a distance, and it was triggered, or triggered some emotions at the moment that the owner of the animal decided to return home.
Okay.
So things like that, and that's interesting. So yeah, you mentioned Rupert Sheldrake. I know him personally, but not very well. I had dinner with him once in Brussels when I was on my second sabbatical in Belgium. So interesting, morphic fields. It's an interesting concept that we are, I guess, that I don't know if even involves quantum effects or some other field. Maybe it's just a biological field, but I think it would be interesting to consider this from the quantum physics point of view. I would say it's somewhat consistent if we can consider our living system generating a quantum field through our mind, which extends. Who knows how far?
Well, any field extends to infinity. It's just it just might be valued near zero, really far out.
Right. But can be entangled with another field, and that's where things get interesting.
Mm-hmm. Yeah. Anyways, I was going to finish sort of talking about drug design and I guess the environment of the university
Right.
for that. And I was curious, do you think, I guess, a brief overview of the actual drug discovery process?
Right. Yeah, well, it's a long process, as you probably appreciate. It takes now up to 14, 15 years from the idea to the clinic, and it typically takes at least $1 billion to create a drug entity which is successfully implemented in the clinical setting. But probably the first 2 or 3 years are currently spent on basic research, which requires sort of atomic level understanding of the target, typically a protein or enzyme or DNA, RNA, and you're familiar with this, and then trying to find not only the target, but target and its partners, because in biology nothing is in isolation. So these targets interact with other proteins, forming networks. So you need to know how they interact, where they interact, and which interaction you want to block, typically. Sometimes activate, but mainly block. And the next step is to characterize this binding domain, the area where you would like to find a small molecule, typically, which would block the interaction. And that's what we actually spend most of the time using computational resources and mathematical physical models of drug-target interactions. And probably the best way of doing it is to do something called virtual screening. So you have a huge library of chemical molecules of different shapes and sizes from different families, and use brute force and try to find which ones are predicted to be the most